analysing data on race, ethnicity, and country of birth, which is the most common measure in Australia, in ageing research and health research more generally. And then Arjun's going to present his work that he's been doing looking at risk factor research in dementia, focusing on all of these different issues that I'm mentioning. So the first one I'm going to talk about, I think most people would be very familiar with, and that's the problem of our research in the ageing space not being representative, and in particular excluding participants from research most often because of their proficiency in English. And this is some work I did a number of years ago that looked at this in terms of clinical trials in Australia, but it's also equally as important in cohort studies, and particularly in cohort studies of ageing. And this is because not being able to speak English well is much more common in older than younger migrants. And so in this slide on the left, you've got the breakdown of Australia's older population, looking at the proportion that are of Aboriginal and Torres Strait Islander background, and then the proportion that are migrants, and migrants from main English-speaking countries like the United Kingdom, for example, or migrants that are from culturally and linguistically diverse backgrounds. And what you can see when you compare that population on the left to all of the cohort studies is, with the exception of the Koori Growing Old Well study, which includes Aboriginal people in New South Wales, and the Concord Health and Ageing in Men project on the far right that had a really large sample of Italian migrants, most of the other cohort studies of ageing in Australia include people that identify as Aboriginal and Torres Strait Islander, and also particularly called migrants. So they're usually quite inclusive of migrants, say, born in the UK, but not called migrants, and that's largely as the result of low English proficiency. The other thing that's really important to understand about that is also when you're using data from these cohort studies, the people that are called migrants in those cohort studies are probably not representative of their group, because they're the members of those groups that speak English. And so they often have a much higher education level, for example, than members of the same country of birth group that don't speak English. And so that has a big impact on the findings of our study, where we might be underestimating how common particular risk factors or diseases are, because we're just looking at a very select population of some of those groups. The next thing that I wanted to talk about is something that we don't really discuss that much in Australia, but it's very important in this field internationally, and that's when people find a difference by race, ethnicity, and sometimes even country of birth, and then they fall to this biological or genetic explanation for what they're seeing. And whilst that is a possible explanation, there's really so many other differences by race and ethnicity that are probably going to be much more likely explanations of health differences. And that includes differences in risk factors such as education and diet, and also experiences of structural racism that might actually be the underlying causes of those differences in education and diet. The other thing that's really important to think about is that your genetic ancestry and your self-identification with a particular ethnic group don't always agree. And so one is not a measure of the other. And I'll give an example of that on the next couple of slides. And the final thing to understand about genetic influence on health is that they're not completely separate from environmental exposures. So your genetic differences and environmental exposures interact. So it's possible that someone from a particular ethnic group with a certain combination of genes that's born overseas might have different risks to someone that's born in Australia with the same genetic background, because they've had different environmental exposures. So this is one of the few studies that's tried to look at the complexity of genetic ancestry and ethnicity. This is a study in the United States, in New York, called the Washington Heights Inward Columbia Aging Project. And for fans of Hamilton and Lin-Manuel Miranda, his previous musical In the Heights was actually set in this region of New York, Washington Heights. So if you've seen that musical, you'll understand that this is a very diverse region of New York. And so the sample that they recruited for this cohort study has a lot of diversity. And the three groups included are non-Hispanic white, non-Hispanic African American, and then the largest group of participants are Hispanic Americans, and they're mostly from the Dominican Republic in this particular study. And so one of the publications from this study grouped their sample into 3 genetic ancestry groups, which were Caucasian or predominantly European, African, or this mixed group that they called admixed Hispanic, which were people that they couldn't really group into the predominantly Caucasian or predominantly African category. And this sort of shows the complexity of trying or I guess what's wrong about assuming that ethnicity represents someone's genetic ancestry, because it shows that although there's a correlation, it doesn't always match. So, for example, there's 3 people that I've identified as white, whose genes are predominantly African. There's 13 people that identify as Black, whose genes are predominantly Caucasian. And then there's quite a number of people identifying as white and Black who have this admixed genetic ancestry that's kind of equally Caucasian, equally African. And if you look at the Hispanic group, for example, there's participants that identify as Hispanic that have all 3 different types of genetic ancestry. So it's really emphasizing the point that race and ethnicity are not biological categories. They're not based on genetic ancestry. And if you have hypotheses around genes, then you should measure genes. You shouldn't be assuming that you've somehow captured genes because you've asked someone what their ethnic identity is. Okay. The next point that I wanted to talk about, I hope I can explain well because I think it's really important, but I remember the first time I started to think about it, I found it complex. And that's the problem that arises when you just adjust for race, ethnicity, or country of birth as confounders. And so perhaps you've tried to do the right thing, you've been really inclusive in your study recruitment, you've got a really diverse sample, you've measured race and ethnicity, and then when you go to analyze your data, you decide to adjust for that as a confounder. So to help think about this, and I would really recommend reading this article, there's a great commentary by an American epidemiologist, Kamara Jones, written more than 20 years ago in the American Journal of Epidemiology, where she talks about this issue. And she gives a hypothetical scenario where you have an imaginary city called Metropolis, an imaginary disease called disease X, but for some reason there's inequalities between the east and the west side of the city in terms of how common disease X is. And then she said you can really choose 2 different approaches to trying to understand disease X. So in the first one, what you're interested in is why there's a big difference between the east and west side of the city. So you're trying to understand what's driving the inequalities in disease X. In the second approach, what you're doing is you're trying to understand what causes disease X. And perhaps there might be differences in risk factors between the east and west side of the city, and you don't want to hide any of the relationship between those risk factors and the disease, so then you adjust for those east-west differences by adjusting for that as a confounder, or perhaps you just do your study in the east or just do it in the west. So these are two very different approaches to understanding disease, and it's actually the second one that we pretty much always use, and that's why we adjust for race and ethnicity as a confounder. And on the next slide, I'll talk about why that matters. And it matters because it really doesn't help us to try and reduce inequalities in our society. So if we take dementia, for example, because that's what Arjun's going to talk about, if you can imagine that there's an ethnic group that has a higher rate of dementia, and then they also have a particular risk factor that causes dementia, such as diabetes, that's more common in that ethnic group. What that would mean if we have a population intervention that's targeting diabetes, that would reduce dementia, and it would also reduce inequalities in dementia because it's targeting a risk factor that's more common in that group that's more disadvantaged in terms of dementia. However, if on the other hand, we have an ethnic group that has a higher rate of dementia, but has a risk factor for dementia that's less common, say, for example, this time depression, if we target that risk factor, yes, we'll reduce dementia at the population level, but we will increase inequalities because we're actually targeting a risk factor that's not an important driver of dementia in that disadvantaged group and is probably going to be an important driver in other groups where dementia is less common. And so this really emphasizes the point that when you use approach 2 and you just look at risk factors rather than trying to understand drivers of inequalities, you might target risk factors that, sure, decrease that disease, but they might drive up inequalities. And so really we need those two pieces of information. We need to use both approaches that Kamara Jones accepted. And it's approach 1 where we don't just adjust for race and ethnicity as a confounder, we try to understand what's driving those differences. That gives us the information we need to target inequalities. The next 3 issues are more simpler, so I left the easier bit to the end. Okay. So the next point that I wanted to talk about is it also seems to be really common in research that involves race and ethnicity as opposed to other areas, and that's the problem of overinterpreting stratified results. And whilst it can be really useful to break a diverse sample into different subgroups, it's just really important to make sure that we don't overinterpret any differences that we see. So I'll just provide an example to explain that a little. So this is a study looking at risk of dementia, and it's a study from the United States, and so they have African American and white participants in the sample. And if you look at the result that I've underlined there in red, you can see that it's showing that in both African American and white participants, that the risk of mixed Alzheimer's disease is much higher in people that have both diabetes and a particular genetic variant, ApoE. And you might also notice that although the point estimate at 5.9 is a bit higher than 2.9, the confidence intervals in the brackets are really wide because this is quite a rare disease, and they overlap with each other totally. And so what that means is that there's actually no evidence from looking at these subgroups that these risk factors work any differently in African Americans than they do in white participants. They're both important risk factors. However, you might also notice that in white participants, this confidence interval here, it includes the value of 1, which means that this result on the right is not going to be statistically significant, whereas this one on the left is. And so the authors in this paper interpreted that as being that these risk factors were important in African Americans, but not in white participants. And that's not correct, and unfortunately is quite common. And so this is really just to emphasize that if you're looking at differences by race and ethnicity, and you've divided your sample into subgroups, you shouldn't be comparing the p-values between those subgroups and then saying that they're different to each other. You need to actually do a statistical test for interaction. You actually need to test statistically the difference between those two groups. And if you think about it, if you have, for example, a risk factor that you find to be a significant predictor of an outcome in your whole sample, when you divide that sample into little groups, some of the groups, because they're smaller, that significant relationship will disappear. That doesn't mean that the risk factor isn't important in that group. It just means that that group is smaller. You don't have a lot of outcomes happening in that group. So you don't really have the statistical power to find a significant relationship. And it can actually be really harmful to interpret that as saying, oh, this risk factor isn't important in that population, as that can lead to people perhaps then saying, oh, we don't need to concentrate on targeting that risk factor in this group. They shouldn't be a priority for intervention. So it's really important, in fact, that these kind of relationships aren't misinterpreted. So the next point that I wanted to touch on, again, is really about how we interpret differences that we see. So perhaps you've done a study and you've found that there is a difference by ethnicity, and then you're thinking about what that might mean. And one of the really important considerations when we find differences is to make sure that difference isn't just from bias in the way that we've done our study. It is actually showing a real difference. And Arjun will talk about this a little bit more, but we really found that most studies don't sufficiently report about this or think about this when they're interpreting results around ethnic differences. And so some questions to ask yourself when you're seeing these kind of differences is to think about whether the way that participants were recruited into the study, if that differed by ethnicity, if response rates were the same between ethnic groups, whether missing data or loss to follow-up differed by ethnicity, and that might include losses due to mortality being different between groups. And then in terms of thinking about measurement bias, it's important to think whether the same method was used for assessing the risk factor and the outcome by ethnicity. And even more importantly, some outcomes have a subjective component. For example, it might involve interpreting a chest X-ray or a CT scan, or it might involve a clinical diagnosis such as dementia, which includes a bit of judgment by the assessor. And so when there's ever any outcome that includes some kind of judgment or subjectivity, that should be ideally done blind to the race or ethnicity of the participants if you don't want bias to be introduced. And finally, if any of the assessments involve language, then and you are including people whose first language isn't English, then you need to consider that in terms of bias and may need to use translated, validated instruments or interpreters. And the final point that I'll make before handing over to Arjun is one of my pet hates in this field, particularly in Australia, and that is the issue of combining multiple diverse groups together. And to give you, and I'll just give you an example of that to really emphasize why it matters. So in the study on the left, they've grouped the population of migrants into those born in the UK and Ireland, the rest of Europe, all of Asia and other, and have compared them to the Australian-born population. And I've just highlighted in the red box the results for cardiovascular disease. And you can see that the rate ratio presented is all below 1. So it means that there's lower rates of coronary heart disease and stroke in all of those large migrant groups. And this study interpreted this as being, oh, this is the healthy migrant effect. Migrants are healthier than the rest of the population. And those kind of conclusions really drive this idea that it's okay to exclude migrants from research in Australia because they're so healthy that there's really nothing to worry about. The study on the right, on the other hand, groups the population into much smaller groups. And here you can see, and they've looked at a similar outcome, acute myocardial infarction. And here you can see by dividing the population into smaller groups, there's actually several groups that have much higher rates of heart attack than the Australian-born population. And that was totally missed on the study on the left because of the way they combined groups together. So I think I'll stop there, and now Arjun is going to present his work on dementia risk factor research that really talks about many of these issues that I've just discussed and gives some specific examples.
Thanks, Fiona. Basically, over the last 2 years or so, Fiona and I have been working on this project, looking at how racial and ethnic minorities have been represented in dementia risk factor research, and we're almost at the end of it now. So we thought we'd share some of the important results that we found through the project. And what you'll actually see is that a lot of the points that I cover here were outlined in Fiona's part of the talk as well. So basically, dementia itself is obviously becoming a major global health problem, with populations all around the world generally living longer. And the World Health Organization's projection is that the current worldwide prevalence of dementia, which is somewhere in the range of around 47 million, based on data from a few years ago, is that it will double by 2030 and probably triple by 2050. So it's all the more important that we actually consider how we do research in this field and consider how that research is going to inform public health interventions in the future. There's also lots of parts in the world which have an increasingly ethnically diverse aging population, Australia being one of them. But unfortunately, the inclusion of ethnic minorities in longitudinal research hasn't really been a major consideration previously. So it's really unclear, firstly, what the burden of dementia actually is in those minority populations. And secondly, it's also unclear how differences in risk factors might be contributing to differences in dementia burden within populations. And so with that in mind, we essentially set out to perform a systematic review that looked to quantify how exactly ethnic minorities have been represented in cohort studies of dementia risk factors, ultimately so that we could identify where there are barriers that could potentially be addressed so that future research is more inclusive and more equitable to everyone in the population. So to make our systematic search more efficient, we actually split our search into two separate parts. Initially, we searched for systematic reviews using those databases from the inception to July 2020. And through that, we found about 3,100 systematic reviews, which we screened and looked through to find individual cohort studies. And then the second part of our search used the same databases, but this time looked at the primary cohort studies themselves from 2014 through to 2021. And ultimately, we ended up finding 97 cohort studies, which we included in our analysis. And so in terms of the most common reasons we excluded certain cohort studies, we firstly looked at how dementia was diagnosed. And we only really included studies where it had been based off of some form of clinical assessment using validated criteria, which was most often the diagnostic and statistical manual used in psychiatry. And for the linkage studies, it was a bit different, but we mainly looked at whether or not dementia case ascertainment was based on both inpatient and outpatient records. We also only included studies that were population representative. So, for example, we didn't include studies which were looking at dementia incidence in samples with a specific disease or a specific risk factor profile. And then finally, we only included cohort studies which had at least 3 publications that we'd found in our search, so that our findings were mainly addressing the larger and more influential cohort studies in the field. All right, so I'll just spend a little bit of time walking you through the framework that the framework and the steps that we use to break down the cohort studies that we had looked at to derive our results. And so the first stage here is basically looking at whether or not ethnicity was discussed as part of the study's selection process, or whether it was described as part of the sample characteristics. And then the second stage below is looking at whether or not ethnic minorities were included, and if so, whether or not they were excluded or under-included through different processes. So in terms of direct exclusion, we looked at whether or not the inclusion or exclusion criteria actively disadvantaged or disqualified participants based on ethnicity or variables that are pretty closely related to it, such as citizenship or country of birth. And in terms of under-inclusion, that was most often related to whether language proficiency was used as part of the inclusion criteria, because that can obviously disadvantage minority participants who may not necessarily be fluent in the dominant language and would therefore be unable to participate. And then the third stage is really where some of our most important findings come from. And so this is where we looked at the extent to which ethnicity has been considered in terms of, in the studies which included ethnic minority participants. And we looked at things like whether or not ethnicity had been measured appropriately. So for example, self-reported ethnicity is often considered the gold standard. And we also looked at how ethnicity has been considered in data analysis. So for instance, was the incidence of dementia or the prevalence of risk factors stratified by ethnicity, or were studies just simply adjusting for ethnicity in their models? And Fiona already touched on that, and I'll discuss that a bit further towards the end as well. It also looked at whether there was evidence of selection and measurement bias, and again, a lot of that essentially looks at the points Fiona talked about a bit earlier. And then finally, we looked at the results of the studies as a whole, including the way they had incorporated race and ethnicity into their analysis to see if the studies produced results that were specifically relevant to ethnic minority groups. So as I said before, we ended up finding 97 cohort studies from around the world looking at dementia risk factors, and as you can see from the table here, the majority are really coming from North America and Europe, together making about 75% of the studies that we found. And the largest cohort studies as well, just in terms of the number of publications we found, also came almost exclusively from North America and Europe. And so a lot of the evidence in this field actually relates to those specific populations. All right, so this first map essentially represents the parts of the world where the 97 cohort studies that we found actually come from. And as you can see, there's not much research coming from developing parts of the world, particularly South America, Africa, and parts of Asia. And then the second map here looks at which countries have cohort studies which report the ethnicity of their sample population. And you might have noticed that Canada's dropped off here as well as parts of Europe and China. And so overall, only about 40% of the cohort studies we found actually reported the ethnicity of their sample populations. And so in contrast to the previous map, the countries here are where cohort studies have reported ethnicity. And have actually included ethnic minority participants as well. And altogether that comprised about 29% of the studies that we found. And you can see that all of Asia is now essentially dropped off as well. And ultimately it's clear that parts of the world where there are significant ethnic minority populations are seemingly underrepresented in this kind of research. And now this last map just essentially serves to highlight that only the US really has produced cohort studies which are specifically useful for understanding ethnic inequalities in dementia. And these North American studies, there's just essentially 7 of them. And the most useful data is actually coming from the Washington Heights Inwood Columbia Aging Project in New York that Fiona talked about a bit earlier. And that looks at differences in dementia risk factors in African American and Hispanic populations. Australia obviously is notably absent here, as well as the UK and Canada, which also have ethnically diverse populations. So it's quite important to note that there's a lot we don't know about ethnic minorities and the burden of dementia within those countries. So next we're looking at a bit more of a granular level at the 28 cohort studies which have included ethnic minorities in their sample. And so altogether we found 394 publications coming from those studies. And of the publications that included a diverse sample, we found that the vast majority, about 90%, considered ethnicity as a confounder. And of those publications analysing race as a confounder, about half actually reported that there was a difference in the prevalence of risk factors between different ethnic groups. And about a third of the publications that analysed race as a confounder actually reported a difference in the incidence of dementia as well by race. And I'll touch on the importance of those findings a little bit later, but it's just important to see that despite a sizable proportion of studies actually recognising race and ethnicity as having important relationships with the prevalence of risk factors and the incidence of dementia, the way that ethnicity is handled as a confounder means that those differences aren't always scrutinised in a way that helps show where and why disparities might exist. And so and of all the publications we looked at, only 43% were useful in understanding risk factors within ethnic minority populations. And probably most concerningly, only 5% of the publications we looked at were actually useful for understanding ethnic inequalities. And as I mentioned before, these publications were largely from a single study, which is the YCAP study that we talked about before. So now I just wanted to spend the next few minutes going through some of the key findings from our results that need to be taken into consideration in future research. And so firstly, we really need to think about the way ethnicity is characterized, and Fiona obviously talked about this as well before. And what we found was that a lot of studies that included ethnic minority participants tended to dichotomize the study population into the ethnic majority, which is most often white, and a combined minority or other or non-white population group. And so that way of categorizing ethnicity is useful in interpreting risk factor and outcome relationships for the majority populations, because it removes heterogeneity, but it doesn't really provide meaningful results for the minority groups where you've got diverse risk factor and outcome relationships, which are just combined into a single category. Another thing to note is that sometimes combining different ethnic minority groups into one category might be the only option if there aren't enough participants in those individual ethnic groups to sufficiently power stratified analysis. And so that needs to be taken into account in the recruitment phase of studies, potentially even by oversampling ethnic minority populations to make sure that analyses using more specific categories can actually be done. And again, Fiona touched on the important considerations that should really underpin our assessment of selection and measurement bias. But I've just included them on the table here as a reminder as well. In terms of selection bias, we found it really difficult to make an assessment of whether selection bias was present because the information we needed to make that judgment wasn't really available in the majority of instances. And really only 6 studies, or about 20%, reported what we needed to know to make that overall assessment. And in terms of measurement bias, again, not all studies actually reported the information that we needed. So about half, or just over half, reported enough information to make an overall judgment. And of the 28 studies, which included ethnic minorities, only 5, or about 20%, had a lower risk of measurement bias. And in terms of dementia diagnosis, the studies which we deem to have a low risk of measurement bias often used translated neurocognitive screening and diagnostic tools, or made sure to have study personnel who are fluent in the languages that participants spoke, such as Spanish in the North American studies, for instance. But very rarely did studies report if consensus diagnoses of dementia were actually made blinded to race and ethnicity. So the last point that I wanted to touch on is how the majority of cohort studies, which include ethnic minority populations, incorporate ethnicity mainly as a confounder that's adjusted for along with other demographic variables such as age, gender, and education. And as Fiona touched on earlier, presenting data on the prevalence of risk factors and the incidence of a disease by ethnicity can give us really valuable information because it can help us to identify if ethnic disparities in a particular disease exist and what the potential underlying determinants of that might actually be. And just to show an example of how that kind of information can be really useful, I've included some screenshots from one of the publications in the YCAP project, which included a large number of African American and Hispanics in the study population. So in this particular publication, it looked at diabetes as a risk factor and actually presented the prevalence of prevalence of diabetes within each ethnic group, which we can see here in the first screenshot at the top. And then using that, the study actually goes on to report that the population attributable risk of diabetes in each ethnic group. And through that, we can see that the population attributable risk of diabetes relating to stroke-associated dementia was about twice as great in Hispanics and African Americans as it was in whites, which we can see reported in the second screenshot just below. So you can really see how useful presenting data stratified by ethnicity can be, rather than just adjusting for ethnicity as a confounder. And so this kind of data can really help us work out the reasons underpinning inequalities in dementia burden between ethnic groups, and perhaps inform where interventions can actually be put in place to address them. So although our work has kind of identified that we really need more cohort studies to include ethnic minority participants, it's worth noting that there are studies underway which are working towards improving our understanding of ethnic inequalities in dementia. And in Australia, for instance, we've got the Career Growing Old Well study, which has been set up to explore the prevalence and incidence of age-related diseases like dementia in Aboriginal populations in New South Wales. And for the Framingham cohort study in the US, which was originally based on a fairly homogenous Caucasian population, they've had subsequent waves of recruitment, which have included African American, Hispanic, and Asian and Native American participants. So hopefully those cohorts should produce data on dementia incidence in the coming years. But ultimately, what needs to happen in this research space is a shift in the way we conduct research such that we start to consider not only the causes of a disease, but also the causes underpinning inequalities in disease. And so to achieve that, cohort studies might need to look at oversampling at-risk populations based on socioeconomic determinants, or even through use of large linked administrative data sets, which can tell us about the trends of disease in the whole population, and allow for sufficient power to examine specific ethnic groups as well. And hopefully through that kind of research, we can work towards designing and implementing targeted interventions that ultimately might help to reduce ethnic inequalities in the burden of diseases like dementia.
Fantastic. Thank you so much, Fiona and Arjun.